Thanks a lot. So Wata team came all the way from Australia, so we're really happy that they made the trip for us, and they're going to present their solutions.
Hi, everyone. Thank you for having me. Firstly, I would like to thank the organizer, especially Pablo. So this morning, he said he sent 500 emails, then I think 30 of them to me. So I'm actually the advisor of Nguyen. So I email Pablo about how can I be a team of myself. So he said it's fine. Then I about to submit under my name, but the system didn't allow me doing so. So I take my student name, but my student didn't do anything on that. It's me. So Pablo allowed me doing so, but it's because the system is not because I'm cheating or something like that. I emailed Pablo before doing so. So mainly in this challenge to me, it find out what is the organizer try to evaluate us. So arguing a lot among us is about how Pablo evaluate us, not about what the algorithm work well. So here I joined the challenge. This is the first team challenge, and invited by my friend Tukle from South Australia. So it's interesting challenge. So the task is predict 10 most likely location per cell using gene expression. So the challenge here is not using 84 genes, but just a subset of that. So immediately I thought feature selection play an important part here to win the challenge. And because it's not allowed us to use the target to select feature, meaning we have to do some kind of unsupervised feature selection. So, I mean, the two guys in the, when they were in the leaderboard, we're not scared of them because I immediately guess that they are overfitting and they are using Y to get out the feature, to select the feature. So when I see they are top, I believe they did something wrong. So we have to do unsupervised here. Here, this challenge, the crowd truth is there. We know the crowd truth, but we don't know what the organizer evaluate. We know the crowd truth because if we are using all the genes to map to the location, we use that like the crowd truth. We use the expression for 84 genes, and we know immediately where the location the cell is. So here the MCC between the binary expression for the cell and the in situ will be used to get out the crowd truth. The MCC is actually very popular in binary classification. We are using that to evaluate if the binary classification works well or not. So here we are guessing what Pablo evaluates us. Then the first would be the list of genes. So that is our hand because that would be due to the feature selection method we are using. So we just trying some kind of feature selection method. And the second would be the similarity in the expression. This would be measured by the expression for the 20, 40, 60 gene with the crowd tool using MCC. And the third thing, I guess, Pablo will evaluate us is based on the location. So it's the distance between the crowd tool and the predicted. And this is a flowchart for the work we are working for the challenge. So initially, from the data, we will go pre-processing and then go feature selection. And when we got the feature, we run through kind of prediction using that measure. We will go detail, follow. So why pre-processing? So my biology friends saying that the dropout is popular in single cell sequencing currently. So they saying that we should do some kind of imputation. Otherwise, if we just based on that, we may learn out some relationship is not true. So before doing feature selection, an imputation would be used. And here we are using metric, which just published earlier of the year. And when we already got the feature, we go through the feature selection. This is the method one for the challenge, sub-challenge one. And initially, we selected gene. That from the prior domain, we got some gene already there, then that would be in the list. And then we add up the gene based on the gene is not selected. If they are farther with the gene already selected, that will be selected into the pool, the feature selection. And we look that until we got the number of genes depending on the sub-challenge, 20, 40, 60. The method two is same with the method one, except for the first stage, we actually don't care the prior. We just use the two gene fight to the rest. That is like the manual selected from 84 gene, and we have to do unsupervised. Meaning we don't use the Y for that. We don't use the Y for selecting that. So the same idea implemented by our physicist. He run his idea like this. His idea actually similar to us. To ours, it like we selected the gene. If they are very difficult to be predicted by the other. So again, here I have to like start with if we are allowed to use the Y, then the feature selection would become supervised feature selection method, and with that, those features gain us a good performance in classification will be chosen. But because Pablo didn't allow us to use the Y, then we have to choose among them. So those genes, it's like very difficult to be predicted by the other. We will chosen into 20, into the feature, into the model. And this idea, I mean, like would be time consuming. So in the method, in the paper, do it faster, and we just adapted that into our feature selection. Then, when we got the picture, we again use, if they are like from the binary expression for the gene selected, we try to find out if they're matching with the route true or not, and the matching will be measured by MCC MCC measure. And we observe that the location is not evenly distributed. So the abnormal location would not be selected. So we do some kind of post-processing there. So by MCC, if they are in some abnormal location, will not be selected, won't be selected. So we remove that using LOF score. And that two-stage will be was used to choose the first two location, and the rest 8 location will be choose based on the distance to the two. And this is the implementation. And all of this is available in the website. Yeah, this is the abstract for the work. Yeah, by that I conclude my talk.
Questions?
Yeah.
Maybe we can leave the. Where?
So when you were computing the MCC scores between the binarized versions, the binarized data, did you re-binarize it or use the provided binary data?
Yeah, I'm just using the provided. Yeah.
Okay.
You're using the provided binary data?
Yeah.
But maybe I'm wrong. I think they binarize the data based on. So somehow when they binarize the data, they're using some structure that they need the feature of the original 84 genes. So
Yeah, as long as you don't use 84 for that, it's fine. So you just use 20, 40, 60, it's fine. It only discussed there. Yeah.
you re-binarize the data.
We don't need to re-binarize. But it's already there as long as you don't use the whole 84 binary to compute the MCC. It's fine.
No, I mean the thing is that I think they binarize that 84 genes data. They're using that 84 information. So they're using that the correlation, like how strong the correlation it is to finalize the data. So if you're using 60 genes, I think still using the same algorithm, you will have a different finalized data. Maybe I'm wrong, just.
What I remember is that they did for each gene, they did a distribution of expression, and they tried to put a threshold, but independent of the 84 genes, that you can binarize all the genes, right?
Yeah, but the binarized data, we assume,
Professor,
yeah.
clarify.
I think what you mean is that there is the resulting binarized data.
Yeah, the
is
result
based
is-
also on the 84 genes information to map the genes properly.
The result is drop-seq data.
Yeah.
When the
so
drop-seq data
when
is
they-
binarized in DistMap, which is what was provided,
Yeah.
it requires the 84 and C2 genes to
Yeah.
generate that binarized.
That binarization. Okay,
so
maybe.
when they automatically,
Yeah, but the binarized data were provided as a file.
Yeah, it was legal, so to speak. But it's possible that there is a leakage there.
Yeah, 'cause
but when
I'm
we
pretty
decoding
sure
your
in the
function,
instructions it said you couldn't do that.
yeah,
The binarized data is provided, but you couldn't use the drop-seq binarized. Just the in C2.
Pablo, do you remember that?
We gave the binarized in situ data. And, I think that's now really, not sure if I correctly binarization of the in situ data is independent of the number of genes.
Yes, of the in situ data,
Yes.
but the drop seq, because if you're computing an
Because you
MCC
based on that
score,
84 genes. If you only based on 20 genes, your binarization will be still the same algorithm,
Yes.
but you will have different binalized data.
No, the binarization of the in situ data is always the same.
yes,
not
but
not
it's the
for in situ. I
binarization
mean
of
for
They-
your
The
drop seq
sequencing
data,
data, that's
the
a
drop
different
seq
question.
data.
They
it
say
should be
that-
different.
They say that in order to binarize the RNA-seq data, you use information pertaining to the in situ. And
Yeah.
in this map.
in this map, but this
Well, how did you then binarize? How did you binarize the RNA data?
can we suspend this until the next talk? Because let's go to the science paper and determine that. My recollection is that there was a gene by gene threshold that was independent of the in situ, if I remember correctly, the gene. But there is a figure there. We'll look at it.
I think the in situ binarization has nothing to do with the drop seq binarization.
Yeah, I think so too. Let's suspend this for one, and we can go to the paper. So the next speaker is, who's the next speaker? There.